This lecture is going to be on glucose. Islet cells of the pancreas produce substances that regulate glucose metabolism, and these hormones are glucagon, insulin, and somatostatin. Glucose can be obtained from the diet or produced by various cell types. While many cell types can perform gluconeogenesis, only the liver can export glucose into the blood, and this is why liver failure can cause hypoglycemia. Of the hormones that regulate glucose concentration in blood, there is only one hormone that decreases blood glucose, and that's insulin. All the other hormones, including glucagon, cortisol, growth hormones, and epinephrine, cause an increase in blood glucose. Blood glucose concentrations may be transiently or persistently increased or decreased. Causes of transient hyperglycemia or hypoglycemia are listed in the first row, and causes of persistent glucose dysregulation are listed in the second row. Most of these causes are caused by increases or decreases in the hormones that regulate glucose. So, for example, cortisol causes transient hyperglycemia due to stress, but persistent hyperglycemia can be seen with hyperadrenocorticism. Many of these causes have already been discussed in previous lectures, so those causes won't be duplicated here. Also, endocrine diseases of the thyroid and adrenal glands will be reserved for future lecture topics. This lecture and the next lecture will focus on glucose disorders that are not discussed elsewhere, or those that are inherent to insulin or glucagon dysregulation. Postprandial hyperglycemia occurs in monogastric animals within minutes to hours after ingesting a meal. Other postprandial changes that may occur include increases in serum cholesterol, triglycerides, bile acids, and ammonia concentrations. High concentrations of triglycerides can also give the serum a whitish color, and that's known as a lipemic sample. Acute pancreatitis can cause hyperglycemia by 3 different mechanisms. Dogs with pancreatitis are often stressed, which can cause hyperglycemia secondary to cortisol release. During pancreatitis, the injured acinar cells release a large amount of glucagon and can be rapidly depleted of insulin. So this can cause hyperglucagonemia and hypoinsulinemia, which results in hyperglycemia. Sepsis causes hypoglycemia due to a hypermetabolic state and an increased tissue utilization of glucose. Support for sepsis includes severe inflammation, as indicated by a degenerative left shift, or evidence of a severe infection, such as a GI rupture. Maternal-associated hypoglycemia is caused by excessive glucose secretion by the mammary glands or utilization by the fetus. So the history is often helpful in making the diagnosis based on finding a lactating animal or a pregnant animal. Neonatal or juvenile hypoglycemia typically occurs in toy or miniature breeds of dogs, and this is due to a low glycogen storage pool in those neonatal animals. Other ClinPath changes include those of young animals, so they might have lower hematocrits compared to adults or higher lymphocyte concentrations compared to adults. Delayed serum separation or plasma separation from red blood cells results in red cells consuming the glucose in the sample. Therefore, this is a cause of in vitro hypoglycemia, so not in the animal. Insulinoma is a neoplastic proliferation of beta cells. These beta cells produce excessive amounts of insulin, and then this leads to persistent hypoglycemia. Other CBC changes, serum biochemical changes, and urinalyses typically don't occur, so often all other tests are unremarkable.